So what I'm trying to do with my research project is improve a methodology for environmental DNA. And what I mean by that is that with environmental DNA, which is DNA taken from the environment, so it's things that organisms are constantly shedding. So that's going to be like skin, scales, fur, hair, feces, urine, all of it. And we take an environmental sample. So in my case, that is a water sample. And then from that, I extract the DNA, and we can use that little DNA pellet to send it off and figure out what species is there. So that's where my project comes in, and I'm asking, okay, so right now what we're storing our samples in is just this plastic storage tube. What if the DNA is sticking to the plastic storage tube, inhibiting our ability to detect our organism? Other manufacturers produce plastic storage tubes that are specifically meant to prevent the DNA from sticking or binding. So if we were to take samples with the normal storage tubes and then the low binding, could we get higher detection rates with the low binding? And I think it's a pretty cool question, because if we are able to get higher detection rates, then it can have good conservation impacts by increasing our sampling efficiency. So DNA, or just using molecular tools in general, has just made the field of biology explode, because there's so many things that we're learning that you can't tell about an organism by just looking at it. And what's specifically really cool about environmental DNA is that we can learn these things about the organisms without ever coming into contact with those organisms. And that's really cool, because when we're talking about endangered species or elusive species, so that's going to be species that are really hard to find, that means you don't have to have direct contact with them, which could potentially kill the organism, stress them out, or it's just very difficult and time-consuming to do that. So the fact that we can take an environmental sample or eDNA sample and derive that or get that information from those samples, and then determine what species is there and their evolutionary history from just a water sample, that's a pretty cool thing. So my research location, it's split between being in the field and then being in the lab. And for my specific project, I'm actually always in the lab because I'm not the one collecting the samples. So that would be over in the Janicek lab in building 58. But for other projects that I help out on, we're at Pensacola Beach taking water samples bi-weekly, and we're coming up on a year of doing that. So it's pretty split right now. I hope that my research project can kind of shed light on if you can get a higher detection rate using a different type of storage tube. And the reason being is that eDNA, you're relying on degraded DNA. So DNA that's, it's subjected to salinity, it's subjected to pH and UV radiation. All these things make DNA break down, and it just makes it that much harder to actually detect the organism that you're looking for. So if there's ways that we can improve the efficiency of the DNA, then I think it would be really beneficial for my project to be able to hint at that and to support, hey, maybe we should look a little bit more into this, so that when we're working with people in conservation, it can be used to improve the methodology of searching for these endangered or elusive species. I'm from this area, from Northwest Florida, so I grew up right next to the Gulf of Mexico. And growing up, having to see the BP oil spill, but also seeing how a town can rely so heavily on tourism and that relationship of exploiting the environment, but also needing to protect it. And then when I took marine biology, I was just learning about all of these very complex issues, such as climate change and ocean acidification. And I knew that I just really wanted to be a part of the solution and research ways to protect biodiversity, catalog biodiversity, and also conserve it.
With my project, I'm going out and collecting samples of water from specific locations of the Escambia River and the Lower Conecuh River. From there, I take those samples back to the lab, where I'm extracting DNA from the water of those samples, and from extractions, I'm able to amplify the DNA with species-specific primers that targets a certain sequence of your target organism, and from there I can test that to see if it'll give me a positive or negative detection. I do a lot of lab work extracting DNA from the water samples that I collect, and that DNA is amplified with species-specific primers, target organisms, sequence of DNA that will lead to a negative or positive detection. Environmental DNA is more cost-effective and time-effective than conventional methods of sampling. It's also less damaging to habitats. You don't need a permit to collect water, but you need a permit to collect the target organism. Environmental DNA allows you to have a better understanding of where your target organism is in the environment that you're sampling. I think DNA is going to give us a better understanding of all the organisms, whether that's aquatic, terrestrial. I think it's going to lead to describing many more new species, especially populations of species that have been seen as one. I think the most positive impact that I want to make with my research is on the population of the river red horse, as well as the understanding of the molecular tool of environmental DNA. In Florida, the river red horse is classified as a species of greatest conservation needs due to drastic decline in its native range here in Florida. This species has been reported as collected 3 times within the past 60 years or so. That raises alarms immediately there. I think with my research, it can lead to a better understanding of how to use conventional sampling methods to target a river red horse in hopes of catching a specimen in the future. I've grown up always fishing. I've loved to fish, whether it's freshwater, saltwater, in any body of water. I love the fact that I can help manage populations of fisheries. I can benefit endangered and imperiled species of fish. And just knowing that I can do that within this field, that's kind of what really made me want to join this field.
One thing that I really loved about UWF, and probably the biggest pull to come to UWF, was that they're so big in undergraduate research.
I think UWF really stresses the importance of giving to the undergraduates.
They really do have so many resources to help undergrads get started.
They want to get undergrads involved,
but also to keep them moving along the way for professional development, being able to do your own research project, getting paid to do research.
I was told that I would develop experience in research and opportunities, and that's exactly what I got.
And then of course, as a marine biology major, I really love being by the Gulf of Mexico and right at the beach.
University of West Florida really gives back to its undergraduate, and I think that was my most important, choice in picking the college, and that's why the University of West Florida was right for me.